So I have the pleasure today of introducing Dr. Stacey Finley, who's giving our talk this afternoon. So welcome, Dr. Finley. Stacey's an associate professor in the Department of Biomedical Engineering at the University of Southern California, and she has a joint appointment in the Mork Family Department of Chemical Engineering and Material Sciences, and the Department of Biological Sciences. So Stacey holds the Gordon S. Marshall Early Career Chair and the director of the Center for Computational Modeling of Cancer. She's also a member of the Norris Comprehensive Cancer Center. Dr. Finley's current research applies a systems biology approach to develop molecular detailed computational models of biological processes related to human disease. And her computational systems biology laboratory develops mechanistic models of biological processes and uses the models to gain insight into the dynamics and regulation of biological systems, synthesize and interpret experimental and clinical observations, provide a quantitative framework to test biological hypotheses, and contribute to the development of novel therapeutics for pathological conditions. And so I'm really looking forward to her talk today on mechanistic modeling of the immune cell activation in cancer. And with that, I'll let Dr. Finley take it away. Thanks very much.
Great. Okay, let me get my slides up. Okay, thanks so much, Morgan, for the very kind introduction, and Cecilia, Dr. Greenwood, for the invitation. It's a pleasure to be able to present some of the work that we've done in my lab at USC, where we are looking at immune cell activation. And so the work that we're doing is within this broader field called mathematical oncology. The goal is to be able to probe the complexities of cancer using mathematical models and computational tools. Cancer is an appropriate context to apply mathematical modeling within because of its complexity. So many different processes. Here I'm just showing angiogenesis, the formation of new blood vessels, different cell types, including tumor cells, immune cells, and then also stromal compartment. And there are processes that are happening on different length and time scales. And so having mathematical models and a mathematical and quantitative understanding of what's happening in the tumor can be very helpful. The main research goal, particularly for my lab, is to be able to predict the dynamics of biochemical reaction networks in cancer. This really stems from my training as a chemical engineer, so I get excited about reaction networks. And we want to be able to understand those networks and predict their dynamics in order to help develop new drug treatments, but also just to understand the effects of existing treatments. And the tools that we're using to do this are systems biology tools. So studying the whole system rather than individual parts. So not just a single molecular species or a single reaction, but how that influences the system as a whole. And we combine mathematical modeling with experimental studies, especially collaborating with experimental research groups. So we're interested in the tumor-immune ecosystem. And so again, it's very complex, many different cell types here, also many different interactions between cells. So certainly tumor-immune interactions, and I'm showing here a tumor cell that can interact with a T cell. T cell can mediate tumor cell killing. Tumor cell can inhibit that and promote T cell exhaustion. There can be interactions between tumor cells and immune and stromal cells, such as how tumor cells can secrete a factor called vascular endothelial growth factor that can promote endothelial cells to form new blood vessels. And then also interactions between immune cell populations. And this is something that I'll talk even more about later in the presentation, interactions between macrophages and T cells. And so what we want to do is to apply mechanistic and data-driven modeling to study cell signaling, to predict tumor growth, and ultimately to explore different treatment strategies. So what I'd like to tell you about today is a tale of 3 scales. And so we'll start by looking at the intracellular scale and looking at signaling inside of CAR-engineered T cells. And then we'll move up to the cellular scale, so examining a heterogeneous population of CAR-engineered T cells. And then finally looking at the tissue scale, so interactions between cells in the tumor microenvironment. And there'll be some points throughout the talk where I welcome questions, but also happy to take questions at the end as well. So CARs, chimeric antigen receptors, these are engineered receptors that can be presented on the surface of immune cells, including T cells and also natural killer cells. And they bind to and recognize a tumor-associated antigen that's presented on the surface of tumor cells. That mediates this activation of a signaling network that can lead to the secretion of cytotoxic factors and cytokines. And this binding is made possible because the chimeric antigen receptors are, the extracellular domain is derived from a single chain variable fragment for an antibody that's specific to the antigen of interest. And that's combined with intracellular signaling domains that are derived from endogenous T cell receptors. So it's really aiming to mimic endogenous T cell receptor signaling in a single engineered receptor that they can lead to tumor killing and cytotoxicity. Now, CARs have been very effective, particularly in B-cell lymphoma, where there's been removal of the disease, even complete remission in several patients, but there still can be some serious side effects that occur when T cell signaling goes awry. And so some of the limitations of the CAR actually provide motivation for our work. So one is called cytokine storm or cytokine release syndrome. This is an overactivation of the immune system that is thought to occur in part because the engineered receptors do not have the same control mechanisms that are present in endogenous T cell receptors. That can lead to a flooding of the body with cytokines and can be very harmful or even fatal to patients. Another limitation arises when trying to adapt the therapy to other kinds of cancers. So I mentioned that in B-cell lymphoma, its CAR-engineered cells are very effective. They are targeting an antigen called CD19, but that same level of specificity where that antigen is only expressed on the disease cells is not present in other kinds of cancers. So for example, we know that certain subtypes of breast cancer have high expression of a receptor called HER2, but that's also expressed in healthy tissues. And so the CAR can have on target, but off tumor effects, which can also be very serious for the patient. So what's being done to address these limitations experimentally is to try to better control the signal, adding in more co-stimulatory domains to better control the cytokine that is released. We've also been trying to target multiple molecules. So having multiple CARs that would target two different antigens expressed by the tumor cells, where that combination is not present on healthy tissues. And then also looking at targeting multiple antigens and also better controlling the signal with PD-1 and other chimeric inhibitory receptors, which are also gaining a lot of enthusiasm. But these are all sort of pursued in a guess and check or trial and error way, rather than having a more fundamental understanding of the signaling that is activated in these CAR-engineered T cells. And so we think that this is a nice place where systems biology approaches could help us to provide more fundamental systematic understanding of CAR-engineered T cell activation, and maybe even help to design more efficient and better CARs. So my graduate student, Jennifer Roars, the first PhD student that I had in the lab, she graduated a few years ago, really pioneered this area of research in my lab to look at a mechanistic model of CAR T cell signaling. And so this is a model that I'll walk you through. It's representing the second generation CAR, which is FDA approved for B-cell lymphoma. And this CAR includes the CD3 zeta chain, which has 3 ITAMs, immunotyrosine activating motifs, 3 ITAMs, each of those having an important, 2 important phosphorylation sites. And so I just denote those with the circles here. This second generation CAR also has the CD28 co-stimulatory domain, which also has a number of important phosphorylation sites that lend to the activation of the CAR. And it recognizes the antigen CD19. Now, an important player here is the kinase LCK. This is also an important kinase in endogenous T cell receptor signaling. LCK undergoes autophosphorylation. It can also mediate the phosphorylation of different sites on the CAR. And once 2 of the sites on the ITAM are phosphorylated, another protein called ZAP70 binds, and LCK can also mediate the phosphorylation of ZAP70. But all the while that these activating events are happening, the cell is also recruiting phosphatases such as SHIP1 to attenuate the signal and remove those phosphate groups. But given strong and durable binding of the antigen, the cell can overcome that negative feedback and lead to the formation of this multi-protein complex called the LAT signalosome. LAT is sort of a scaffolding protein that multiple other proteins can bind to. This is a multi-protein complex that ultimately signals through the MAPK pathway, leading to activation of the transcription factor ERK, which we know is important for promoting cell proliferation. Now, ERK actually provides positive feedback because it can mediate phosphorylation of LCK at a protection site that prevents the dephosphorylation by the SHIP1 phosphatase. And then we also have some strategies or mechanisms by which CD28 influences and augments this signaling through the binding of different adapter proteins such as GRB2 or GADs. Now, this is a complex network, right? There's positive feedback, there's negative feedback, there's this multi-protein complex that happens to form. It's actually a simplified schematic, right? Because I'm not showing all of the bimolecular interactions, but still it's not clear how, for example, changing the structure of the CAR, adding or removing this costimulatory domain or mutating sites in CD3 zeta would influence downstream signaling leading to ERK. And so that's where we are applying mathematical modeling to better understand the signaling network. So just a few details about the model itself. We use BioNetGen. This is a rule-based modeling tool that allows us to automatically generate the reaction network. And so just to give you a sense of what I mean by this, we know, for example, that GADS binds to a site on CD28, and we can define that biochemical rule. And that interaction is happening independently of what else is happening on the CAR structure. So whether or not ZAP70 is bound, whether or not all the ITAMs are phosphorylated, GADS interacts with CD28 in this way. And so we define a subset of biochemical reaction rules, and then BioNetGen implements all of the permutations of those rules. And so it gives us then a set of differential equations, ordinary differential equations that describe how the species concentrations evolve over time. Now, of course, with any mathematical model, we have parameter values. And so we estimate the values of the parameters in our model by fitting the model to experimental data, wherever the parameters have not already been measured individually. And we do this using particle swarm optimization. This is a global learning algorithm that allows us to match the model outputs to the experimental data, and then in the process of doing that, estimate what the optimal parameter values are. So let me tell you some of the results. We started with great ambition and just put together this mathematical model. We tried to fit it to some data that my student Jen collected, flow cytometry data for ZAP70 phosphorylation, ERK phosphorylation, PLC gamma, and others. And that failed pretty miserably. There was just too much uncertainty in the estimated parameter values, not enough experimental data to really be able to calibrate the model properly. And so then we took a step back and instead pursued a bottom-up systems biology approach where we put together submodels. And so the first submodel that we put together, and then we can combine them all together, is to look at LCK activation. Again, LCK is this initiating kinase that promotes downstream signaling. It itself has a program that confers catalytic activity involving the inhibitory kinase CSK and the phosphatase CD45. And so we put together a sort of minimal model of this using some data from Ronald Vale's group that was published in 2014. We put together the model. We were able to predict the LCK catalytic activity, which hadn't been measured experimentally, and also provide some explanation about how its catalytic activity is controlled. And so that's the very first submodel that we published a few years ago. And then we started marching down the pathway. So then we said, now that we know the catalytic activity of LCK, can we predict and understand the rate at which it phosphorylates different sites on the CAR structure? So prior to our work, this really hadn't been explored in great detail. And so my student Jen combined both experiments and modeling to look at LCK-mediated CAR activation. So the experimental system that she implemented was again from Ronald Vale's group, where they used an in vitro liposomal membrane system to represent the membrane environment of T cells. And then on the membrane, she can express different proteins. So she expressed CAR, the CAR construct, she also expressed LCK. And then she could add ATP for different amounts of time and quantify the amount of phosphorylation on the CAR using phosphoproteomic mass spectrometry. So the data that she collected are the circles here. I'm showing the amount of phosphorylation of the 6 different sites on CD3 zeta as a function of time. And so the sites are colored according to which one we're referring to. The lines are the fit of our model. So you can see a very nice correspondence between the experimental data and what the model would predict for the amount of phosphorylation as a function of time. Just a few checks that we did is to say, well, just using the experimental data, what would the half maximal time be? So how long would it take for each of those 6 sites to reach 50% phosphorylation? And so if we just use the experimental data, we would get these half maximal times that are indicated by the black circles here. And if we take our mathematical model and predict what the half maximal time would be, those would be the bars. So again, we see a nice correspondence between the model predictions and the experimental data. We also asked the question about whether there's any cooperativity between the phosphorylation sites. And a way to characterize that is using the Hill coefficient. And so if the Hill coefficient is around 1, then the sites are really operating independently of one another. Again, we can see what would the Hill coefficient be if we just took the calculation from the experimental data using the circles, that's what's shown, or if we use our mathematical model, and that's what's shown in the bars. So again, a nice match with model and prediction. Now, this is just for the CAR that only has CD3 zeta. Then we said, well, what happens for CAR phosphorylation if we add the CD28 co-stimulatory domain? So what we looked at is, again, measuring the amount of phosphorylation as a function of time, now with a CAR that has CD28 present, but still looking at the phosphorylation on CD3 zeta. And so what you maybe noticed is that all of the circles, and then of course our corresponding model fits, shifted to the left, which means that the presence of CD28 enhances the rate at which the sites on CD3 zeta are phosphorylated. We can also get at that by looking at the estimated parameter values. So if we look at only the catalytic rate, so the rate at which LCK mediates phosphorylation of the sites on CD3 zeta, when CD28 is not present, that's what's shown in the black hashed bar here. And then when CD28 is present, we see a 3-fold increase in that catalytic rate. We also tried to understand the importance of different tyrosine sites on CD28 by mutating them to phenylalanine, and we can see how they affect the catalytic activity of LCK. But the takeaway message from here is that the presence of CD28, this co-stimulatory domain on the CAR, increases the rate at which LCK phosphorylates sites on CD3 zeta. And so maybe a potential explanation for this is a physical interaction. So the presence of that co-stimulatory domain recruits more of LCK to be closer in proximity to the CD3 zeta sites, and that is a mechanism potentially for increasing the phosphorylation rate of the sites on CD3 zeta. But one thing to note here is that this level of detail really hadn't been explored prior to our work in this area. It was known that CD28 does something. It enhances some aspect of CAR activation. We know that perhaps it led to greater killing of tumor cells or more proliferation, but some of the mechanism underlying those functional responses hadn't been understood. And so this was a novel contribution that we were able to make. Now, that is, again, just for this second submodel, combining it with the first submodel that I mentioned very briefly, and then we can put together a more comprehensive model of CAR-mediated T-cell activation. Now, not just looking at CAR phosphorylation, but also looking at downstream signaling through ERK, the transcription factor. And so what we did now, and again, this sort of shows the modularity of our mathematical model, but what we did next is to ask the question, well, what is downstream signaling and how does the activation of ERK change when we have CD28 present or not? So what is the role of CD28 in activating downstream ERK? So to answer this question, we used our mathematical model and implemented different hypotheses about how CD28 could be influencing ERK phosphorylation. So I mentioned that there are two adapter proteins. One is called GRB2, one is called GAZ. They bind to CD28 and in different ways influence ERK phosphorylation, either through the LAT signalosome or straight through the MAPK signaling. And then we also implemented a third strategy that is taken from submodel 2 that I just presented to you, which is the effect of LCK having a faster phosphorylation of the sites on CD3 zeta when CD28 is present. Okay, so we can implement each of these hypotheses individually and then predict the ERK response time as a function of the level of stimulation. So how much antigen are the simulated cells seeing? And how does that influence the ERK response time? ERK response time is the time that it takes for half of ERK inside of the cell to become doubly phosphorylated in our simulated cell. And so what we did is to make this prediction for the three different settings where we have CD3 zeta only present on the CAR or a CAR that has CD28 along with CD3 zeta. So for the results on the two left figures, for GRB2 binding or GADS binding, we find very similar results, that actually the presence of CD28, which is this red dashed line, slows down the ERK response time. And we see that because the red dashed line is above the blue line when we have CD3 zeta only. So actually the presence of CD28 is slowing down the ERK response time, especially at low antigen concentrations. At high antigen concentrations, CD28 is not having really any effect at all. And that's in stark comparison to the results that we predict when we have enhanced phosphorylation rate of CD3 zeta due to the presence of CD28. And here, for all of the antigen concentrations that we simulated, the CD28 curve is below the CD3 zeta only curve, which means that it's speeding up the ERK response time, so smaller ERK response times. Now, these are all model predictions. We wanted to try to test and validate and discriminate between which would be correct. And so we turned to an experimental system where my student Jen engineered Jurkat T cells to express either a CAR that has CD3 zeta only or a CAR that has CD3 zeta along with CD28. She also engineered K562 cells. So this is another cancer cell line to express CD19. And then combine the CAR cells and the target cells in different ratios in order to change the amount of stimulation. Then she measured with flow cytometry the ERK response time. And so these are her experimental results, where you can see the blue is for the CAR that only has CD3 zeta, red is for the CAR that has CD3 zeta along with CD28. And we see the effect of CD28 in speeding up the response time. So those curves, that curve is below the blue curve. And just to remind you, these are the model predictions. So again, a very nice qualitative match between the model predictions and the experimental testing. We do know that the ERK response time that we predict with the model is much slower than what she measured with flow cytometry. We think that this has to do with the fact that this ODE model is not accounting for the physical interactions. And so we don't have perhaps this physical interaction between the two different between the CAR and the target cells that we're just not able to account for with our mathematical model. But certainly the qualitative comparison is very nice. And so the model helps us to understand the effect of CD28 and how it influences CAR-mediated T cell activation. Again, gaining more mechanistic understanding about why it would be useful to have this particular co-stimulatory domain present on the CAR itself. So we scaled up to the cellular scale, again, focusing on CAR-mediated T cell activation. But now we wanted to ask the question about what is the effect at the population level. Now, the reason that this is important is because with CAR T cell mediated killing and this type of cell-based therapy, it's adoptive T cell therapy. So what that means is that the patient's very own T cells are extracted from their body. They're engineered ex vivo to now express the engineered receptor, and then this population of cells, usually on the order of 10 to the 6 or 10 to the 7, is given back to the patient. So there's inherently some variability in that population of CAR engineered T cells. And so we wanted to know what would be the effect of that variability. So this is work that my student Colin picked up following on Jen's work to simulate a population of heterogeneous CAR engineered T cells. So again, we're looking at the amount of phosphorylation of ERK as a function of time. And this is just a representative sample of 100 of his simulated heterogeneous cells. Now, how did he incorporate heterogeneity? Well, he changed the initial concentration of the signaling species. So that's one aspect of cell-to-cell heterogeneity. We can take from literature what the range of the initial concentrations might be, for example, of ERK or of VAP or ZAP70, other species in the model. And we sample from this distribution in order to get a heterogeneous population of 100,000 cells. But here I'm just showing 100 cells just to talk through some of the heterogeneity that we see. So we can clearly see that some cells reach 100% phosphorylation very rapidly. Other cells do reach 100% phosphorylation, but it takes longer. And then there are some cells that never reach an appreciable amount of phosphorylation of ERK. So here we already see quite a lot of variability just by changing the initial concentrations of the species to start the signaling model. So what we did is just to bin these responses into two different classes, whether there's a low ERK response, meaning that phosphorylation of ERK at the end of 15 minutes of simulation time never reached 50%, or a high ERK response, meaning that it exceeded 50% phosphorylation. And just a little bit of justification for these two classifications of the ERK response is because more than 90% of the response is either very high or very low. And so there's very little in between. And so it's appropriate to have this binary classification of the ERK response. I'll just note that that also happens in endogenous T cell receptor signaling, that there's either a strong ERK response or very little phosphorylation of ERK. So what we then said is, we know what we changed in the model, right? We changed the initial concentrations. We have this classification of high or low ERK response based on our model simulations. Can we predict whether the cell would go towards a high or low ERK response just based on the initial concentrations? And so to answer this question, we use partial least squares regression analysis, or what I like to call machine learning light. It's just regression analysis to relate the inputs to outputs. Our inputs are the initial concentrations that we vary in the model. Our output is this classification of high or low ERK response. Part of PLSR is just a principal component analysis. So we can very clearly see a separation of high ERK responders versus low ERK responders based on this first component, which accounts for about 90% of the variation in the model simulations. There is some scatter in the vertical direction. And so that's something maybe we could go in and say, well, what distinguishes this low ERK responder compared to this one up here? But we can clearly have a separation of the high and low ERK response. And then we can go in and dig into this analysis and say, well, what is driving those two different responses? So part of PLSR is to get out what's called the variable importance of projection score, VIP score. A good rule of thumb is that if the VIP score exceeds 1, then that species is, or that input is strongly affecting the output. So we have just highlighted the VIP scores for those species that are greater than 1. And we see that it includes CD3 zeta, it includes the phosphatase SHP1, and then also some species in the MAPK signaling pathway. Something that's not shown to strongly influence the final ERK response level is the initial starting concentration of ERK. So it really doesn't matter how much ERK the cells start out with. That's not going to influence whether they will go to a high phosphorylation state or a low phosphorylation state. Okay, so this is just telling us which ones are important. Then we can ask the question about how are they important. So now I've color-coded the bars based on whether they shift the cells towards having a high ERK response or a low ERK response. So some of these make sense, and I'm just going to highlight a couple of these, where if we increase the amount of LCK, that's that initiating kinase, if we increase LCK, it will shift the cells towards having a high ERK response, which is why I've colored it in blue. On the other hand, if we increase the concentration, the initial concentration of the phosphatase SHP1, it will shift the cells towards having a low ERK response. So it's colored in red. Now, something that we were scratching our heads about is this other, red bar here, which says that increasing the concentration of CD3 zeta, so basically increasing expression of the CAR, can shift the cells towards having a lower ERK response. And so it doesn't immediately make sense. It's sort of counterintuitive. Why would having greater CAR expression lead to a lower ERK response level? And so we could look into the mechanistic model and answer and explain this prediction. So what I'm showing is a set of curves that have different initial concentration of CD3 zeta, going from a low value here in the bright green to a higher value in the dark blue. And you can clearly see that the amount of phosphorylation as a function of time shifts to the left. So having more and more CD3 zeta expression slows down the ERK response time. And at the same time, it increases the concentration of the phosphatase SHP1. So again, going from a low CD3 zeta concentration and seeing how it changes as you increase the concentration of CD3 zeta, it's slowing down the ERK response time, but also increasing the amount of the phosphatase. And so it's giving us an explanation as to why increasing CD3 zeta concentration reduces the ERK response level. It's because it's enhancing the recruitment of this phosphatase, the negative feedback loop. So the model helps us to understand how having too much activation, namely overexpression of the CAR, could potentially have unintended effects in actually reducing the ERK response. So just to put a short summary here for the intracellular and cellular scales, we constructed this mechanistic model of CAR T-cell mediated signaling. We can use the model to explain how different CAR constructs influence cell signaling and activation, mainly focusing on the ERK response time. And then we combined the mechanistic model with data-driven modeling in order to understand what are the influential inputs here in terms of affecting the ERK phosphorylation level. And by doing that combination of mechanistic and data-driven modeling, we can provide more biological insight into perhaps how to modulate the cell response. So maybe we want to achieve a certain level of ERK phosphorylation or have a certain distribution of ERK phosphorylation. We can use our modeling framework in order to say, well, how should we overexpress or reduce the expression of different signaling species in order to attain that optimal response or desired response. So this wraps up the first two scales, intracellular and cellular. I thought it might be nice if there are questions to pause here before moving on to the cell scale for another maybe 10 or 15 minutes. So are there any questions here that I can answer?
Yeah, there's one that's just come into the chat from Rodrigo about the intracellular scale. Is LCK recruitment a limiting factor? Is there information on the distribution and activation of LCK in live cells?
Yeah, it's a great question. So just to give a little bit more detail, we took the distribution, the ranges of the initial concentrations and their distribution taken from experimental literature. So we do know, or at least have an idea about what the distribution of LCK concentration is. From my understanding and reading of the literature, it's not so much a limiting factor in terms of influencing its availability in the cell. So it's not influencing or having that be the barrier for T-cell activation.
Thanks. Anwar?
So the question I have is about your initial level of ERK. I mean, I'm a little bit, to be honest with you, a bit confused because your simulations seems to suggest that there's bistability in your system. Yeah. But then somehow in your analysis of the data, it seems like it doesn't depend on the initial level of ERK. So how do you reconcile these two things together? And I mean, when I look at even the cluster, like the figure that you see, I don't really see a clear two clusters. So it seems like the model has inconsistency with the data here.
Yeah, so this is the partial least squares. Really, it's a principal component analysis. So I agree that there's some overlap here, certainly in the middle, but we can definitely account for, I think, these extremes of having low ERK response and high ERK response. And again, when I mention, or what I mean by saying the response, is how much of ERK is phosphorylated. So the relative phosphorylation of ERK, not just how much is there. So whether it's reaching 50% phosphorylation compared to the starting amount of ERK. So this bistability is shown to occur also in endogenous T cell receptor signaling, where this signaling pathway can convert an analog signal, a continuous change in the concentration, for example, of CD19 or the stimulation species, into an on or an off. And so it's due to, and you can see it here, there's this feedback, the slowing down of the ERK response. So even as at very high antigen concentrations, there can be a slowing down of the ERK response that also influences the level of phosphorylation of ERK. And so I think that it makes sense that there's a bistability in the system, and that it doesn't necessarily depend on how much of ERK is there, but really, given the amount of ERK that is there, what is the recruitment and the phosphorylation rate of these downstream signaling species?
I think maybe we should move on, Dr. Finley. I know there is one more question.
Sure.
I don't know how much time you have planned in your remaining.
Yeah. Yeah, okay, so I will continue, and then I'll have more time for questions at the end as well.
Okay, great.
So what Colin then went on to do is to try to embed some of this intracellular signaling reactions inside of cells, and then focus on interactions between cells. So looking at interactions between cells, but then also how cell behavior is driven by this intracellular signaling program. And the goal here is to be able to study tumor-immune interactions and really explore the tumor-immune ecosystem. And so we're moving to a different type of model here, an agent-based model that allows us to look at individual agents or entities in the tumor microenvironment. And so the tool is agent-based modeling. The agents are tumor cells, T cells, both inactive and active, macrophages, where they can be naive or towards an M1 or M2 phenotype, which I will describe in just another slide. And also now we have a continuous layer of diffusible factors. So these cytokines IL-4 and interferon gamma, and their concentrations and distribution in space are described by partial differential equations. So let me just give you a little bit more information about the agents themselves. So we have T cells, they can become activated and mediate tumor cell killing. Macrophages can differentiate from a naive state into either an M1 state or an M2 state. M1 macrophages are anti-tumor because they enhance T cell mediated tumor cell killing. M2 macrophages are pro-tumor because they oppose T cell killing. And T cells can secrete a cytokine called interferon gamma, which promotes the differentiation of naive macrophages into M1. And tumor cells secrete IL-4, also macrophages, M2 macrophages secrete IL-4, and that promotes the further, promoting of naive macrophages into this M2 state. So M1 is anti-tumor, M2 is pro-tumor, and then the cells interact in these different ways. We also, as I mentioned, have this layer of continuous variables. I just want to give an illustration of that. So the color, the yellow color here, is meant to describe the concentration of IL-4. So tumor cells and M2 macrophages secrete IL-4, so there might be a higher concentration here, and then it can diffuse away, and that's illustrated by the lighter yellow color. So I mentioned that Colin also wanted to incorporate intracellular signaling. And so what we did is to take a published paper by Zhao from Sasha Poppel's lab at Hopkins. And they have a model that looks at the signaling cascade that influences macrophage differentiation. And so just to give you a schematic of how we incorporated intracellular signaling, I'll show this toy model. So we have an ODE-based model. They have this ODE model for macrophage differentiation. It's a signaling model. We can simulate this ODE model and really do Monte Carlo simulations in order to get a range of signaling concentrations and time courses for different experimental, simulated conditions. And then we have the simulated data set. We can take the output as being categorical. Again, this binary, does it go to M2 or does it go to M1? And the way that it was defined by Zhao et al. Is to just look at the area under the curve for the signaling species that promote M2 differentiation relative to the area, the sum of the area under the curves for the signaling species that promote M1 differentiation. And so essentially we get this M1, M2 ratio, and that tells us whether the cells will go towards an M2 or an M1 phenotype. So we take the simulated data set, we have this categorical output, and then we encode it using a neural network. So the inputs now are the signaling species that influence M2 and M1. The output is the categorical response. We can encode it with this neural network and then embed the neural network into each of our macrophage agents. And so this is the way that we account for intracellular signaling and how the macrophages would take in IL-4 or take in interferon gamma, process it through this neural network, and then make the binary response to go towards an M2 or an M1 phenotype. So what we're doing is running this ODE model hundreds of thousands of times outside of the agent-based model to come up with this simulated data set, and then encoding that with a neural network that we embed into our individual agents. What this gives us is a vast speed up in the computational time because we're not solving this set of 40 or more differential equations inside of every agent. Rather, it's just this very fast computation that takes in the inputs and converts it to a decision about whether it could go towards an M2 or an M1 phenotype. So we can encode intracellular signaling, but do it in a computationally efficient manner. So what can we use this agent-based model to do? Well, of course, we can look at the cell's dynamics, but we also wanted to look at the effects of different immunotherapies. So I'll just show a few of these results. This is a spatial snapshot of the output from a mathematical simulation. We run the model out to 200 days. We can look at where the tumor cells are and how many tumor cells there are, as well as the T cells, mostly here in the inactive state. We have a few active T cells on the periphery of the tumor, as well as mostly M2 macrophages here. But this is just one snapshot at the end of a 200-day simulation. We can also look at the dynamics over time. And here I'm showing the number of cancer cells as a function of time. This is an agent-based model, which means it's probabilistic. At any time point, there's a probability that a cell could do something. And so we need to run the model many different times in order to extract the conclusions from the simulations. And so I'm showing each of the curves as is indicating one simulation. So we did 100 of those. And for 99 of them, the number of cancer cells comes to sort of this equilibrium state where the tumor is not growing or shrinking just due to the endogenous immune system. And there's just one lone curve, I've colored it in red, where the endogenous immune system can eliminate the tumor. So we see this equilibrium state even if we look at number of macrophages or number of T cells, both the total number and the number of active T cells as well. And this is maybe could be considered the immune control phase of tumor growth that happens over months and years. It's hard to capture in vivo models and certainly difficult to capture with human subjects, but it involves the selection of a population of tumor cells that eventually grows resistant to the endogenous immune system and could potentially take off towards having exponential growth again. But this is just, okay, what's happening with the endogenous immune system? We can also ask the question about how does the tumor respond to different treatment strategies. And so 3 that we looked at here are all macrophage-based treatments. We were really excited about this because it allows us to utilize the model and understand the effects of different cell-cell interactions in the model. So we have one strategy to just deplete all of the macrophages in the system. Another strategy is to inhibit their recruitment towards the tumor. And then the third strategy is to re-educate M2 macrophages into M1 macrophages. So converting those pro-tumor macrophages into anti-tumor macrophages. And the way that this happens is through inhibiting PI3 kinase. So that's why it's important to have this intracellular signaling, because it allows us to simulate molecularly targeted therapies. So we can inhibit PI3 kinase in our ODE model, re-encode that using the neural network, and then be able to simulate this re-education of macrophages. So let me just show you a few of our results and then I'll wrap up. We looked at the fraction of tumors that are removed for each of the 3 treatment strategies, right? Again, we're running the model 100 times or more. We can say out of those 100 times, how many of the tumors are eliminated due to this treatment. And we can change the depletion probability or the strength of the treatment and see how effective is it. And so we can see that there's a sharp increase in the effectiveness of this treatment strategy just with a small change in the depletion probability. That also, and then as you continue increasing, all the tumors are eliminated. And we can also see how long does it take to eliminate a tumor. We can do the same for inhibiting recruitment or re-educating macrophages. The takeaway message is just as we, of course, increase inhibition strength at different levels, then the treatment strategy is effective in eliminating all of the tumors. And then also for re-educating macrophages, it can eliminate tumors in a shorter amount of time compared to depleting macrophages or inhibiting their recruitment. We can also look at the composition of the tumor. So not just whether it's eliminated or not, but also how many M1 macrophages, the pro-tumor, the pro-immune, so the anti-tumor macrophages are there, and also how many active T cells are there. So just looking at the immune composition as well. And so the takeaway message from here is that re-educating macrophages is shown to greatly increase the number of M1 macrophages. We shouldn't be surprised by that. That's what the simulated treatment is designed to do. But it has the side effect of also increasing the number of active T cells by an order of magnitude compared to inhibiting recruitment or depleting macrophages altogether. So we can convert M2 to M1. We can eliminate the tumor in a shorter period of time. We can also increase the number of active T cells as well. So I think that I'll show one more set of results, and that has to do with cycling treatment, just indicating that we might not want to only give continuous treatment for 200 days or more, right? That's not realistic. We could have patient compliance issues or just side effects due to the treatment itself. And so we cycle treatment on and off and predicted the fraction of tumors that are removed. The darker the box here, the larger the fraction of tumors that are removed. And what I want you to take away from here is that inhibiting recruitment, when you cycle that treatment on and off, it's really not effective at all compared to continuous treatment, and also compared to the other 2 treatment strategies. And then re-educating macrophages is shown to be very effective across a wide range of settings. So even having a very long cycle duration, so 25 days of the cycle duration, and only having the treatment on for 2 days out of that cycle, even in that condition, nearly all of the tumors are removed. So again, the macrophage re-education strategy is shown to be very effective in inhibiting tumor growth, controlling tumor size, doing that in a shorter period of time compared to other treatment strategies, and also improving the immune composition of the tumor tissue. So with that, let me just summarize this part of the talk. I described to you a multi-scale model of tumor-immune interactions. We were able to encode and include intracellular signaling using a neural network. We could use the model to predict the dynamics of how the cells evolve over time, also the diffusible factors, which I didn't show, but we also have the time courses for those as well. And then looking at how to compare the efficacy of different immunotherapeutic strategies and using the model to understand not only the effect of the strategies, but how we can see the differences across them as well. So just one ending thought is where this is going, right? There are a lot of experimental tools that are being used to profile the tumor microenvironment. Immunoprofiling also over space and time. And perhaps we can use those as inputs to our model. So the starting point for a particular tumor in a preclinical model, or even for patient samples as well. And then we could say, let's start with this distribution and composition of the tumor and use the model to predict the trajectories and also the response to treatment. So really excited to connect even closer with our collaborators to use their data and to begin to answer more clinically relevant questions. So with that, I'll end. I'll acknowledge Jen as well as Colin, our collaborators, and this is my lab a few months ago when we were celebrating the graduation of 3 students, and certainly our funding sources. And I thank all of you for your attention, and I'm happy to take a few more questions.
Thank you so much. That was a wonderful presentation. We have a question from Francois Bourassa.
Yes, hello, thank you for your wonderful talk. I'm sorry to go back to the first part of it. So I have actually 2 small questions. The first one, could you explain in your second sum model where you looked at CD3 zeta phosphorylation, could you explain again why some sites took longer to phosphorylate compared to others? Was that something that you put by hand in the model, or is there something else? Okay, thanks.
No, so let me see if I can go back here. Let's see. So I'll go back. Right, so just from the experimental data, we found that the different sites are phosphorylated in, really it's in a random order. So the experimental measurements show that A1 is phosphorylated first, right? And then B2, and then so on. So what we did with the model is to say, well, this is the experimental data. It's telling us perhaps this defined order. And so we encode it, we did hard code that into the model and say, if we force A1 to be phosphorylated first, and then B2, and then so on, would we recover this these time courses that we measured experimentally? And the answer was no. Like, we could not have a predefined order of phosphorylation. It had to be a totally random order. So we're not hard coding in this model that I'm showing a precise order in which the sites are phosphorylated. So that's another sort of mechanistic question that we could answer with the model. Is there a defined order of phosphorylation? The answer is no. They're phosphorylated in a random order, and we had to have that random nature in order to match the experimental data.
But each site has a different rate, is that correct? Based on fitting the data?
Yes.
Okay, I see.
So each site has, so LCK phosphorylates, so we have a single phosphorylation, like catalytic rate of LCK, but different rates of binding, like affinity between LCK and each of the sites, right? So it binds first and then it mediates phosphorylation.
Okay, thank you. Okay, I think I'll let someone else ask questions and come back at the end if there's still time. Thank you.
Are there other questions right now for Dr. Finley?
I have a question, if that's okay. It's actually on the first part too, Stacey. Sorry, though, I really like that agent-based model. Thanks for the great talk. But I was wondering, actually, in this plot of the intracellular scale, what could account for, or what do you interpret this kind of dip and rebound? Is that just, and sorry, I should say in the ERK response time. Do you think that that's something that's really happening, or yeah, because it kind of showed up in the data as well?
Yeah. So the endogenous T cell receptor curve for ERK response also shows this slowing down of the ERK response time at high stimulation values. And so it has to do with that recruitment of the phosphatases and having that negative feedback loop, so that even when there's a large amount of stimulation, high stimulation, there is a slowing down of the response. So this is seen in endogenous T cell receptor. It was interesting that we also saw it in the CAR engineer, but it makes sense because of the phosphatases.
Great, thanks.
Rodrigo, did you have another question? I just see you turned your camera on. Go for it.
Yes, I guess I also wanted just to thank you for such a nice talk. I really enjoyed it. I was wondering in the last part about the re-education of the macrophages, because the way that I understood it was a little bit, well, my confusion is a little bit, is the re-education converting M2 macrophages into M1, or is it more biasing the decision-making of M0 towards M1? That's the way I understand it because of what you said about the PI3K.
Right, yes. Okay, this is a good point to clarify. So so we assume that macrophages are plastic. So just because it's in M2, it could randomly convert to M1 as well. That probability is very low, but it depends on the intracellular signaling species. It depends on that neural network. So there is plasticity. We update the macrophage states, I think, every 6 hours. So they are updated again because they could be moving around. They could be experiencing different levels of IL-4 and interferon gamma, and they need to reprocess their environment. So we allow for that. But for the simulation of the macrophage re-education, we're saying that we're I guess maybe to be fully correct, I would, probably say we're more biasing the differentiation towards M1, but it's possible because we are also inhibiting PI3 kinase even in M2 that it can promote their conversion to M1.
And would that, so what I was wondering was, what about trying to inhibit the production of IL-4 by the M2 macrophages? Do you think that would have a similar effect that would help like reducing that positive feedback towards like pro-tumor? Maybe that could help.
Right, yeah, so there's that link here, right? Tumor cells and M2 macrophages are secreting IL-4 that further promotes differentiation towards M2. So that could be another strategy to implement with the model. We took the depletion, and I do think there would be somewhat redundant compared to the re-education, but we could also include that one as well. We took this inspiration from some preclinical studies that were being pursued in mouse, and even early stage phase 1 clinical studies for that PI3 kinase inhibition and re-education. There are others, I think, inhibiting part of PPAR2, and that's another PPAR, I think actually it's gamma, and that's another strategy to convert M2 to M1 or further enhance differentiation towards M1. So yeah, I think there are lots of other ways that we can move in this direction, and also consider other aspects of heterogeneity of the tumor microenvironment as well.
Yeah,
I see a question in the seminar room.
Yeah,
You walk over to the speaker.
He's at the mic, Celia.
Okay.
Hi, thanks for your talk. I wanted to ask, do you know what part of your network is responsible for this binary decision for ERK? You either have low phosphorylated ERK or high phosphorylated ERK. I'm assuming that's correlated with the percentage of bound ZEP70 or how completely the ITAMs are phosphorylated, but like what is, do you know which aspects of your network actually are responsible for essentially turning ERK into like a switch that's either on or off?
Exactly, yes. So it has to do with, and you hit it right on the nail, the CD3 zeta and having the the three ITAMs and their them being doubly phosphorylated and then influencing that signalosome formation. So when we think about endogenous T cell receptor signaling, this MAPK pathway also exhibits that ultra sensitivity of either being basically on or off, but it has to do with what's called kinetic proofreading. And that's also something that's incorporated when we have, I'm not showing it here, but have the three ITAMs in their doubly phosphorylated forms, leading to the formation of that lat signalosome. So it's an important question, and it's sort of analogous to some of the observations from endogenous T cell receptor signaling as well.
Thank you. Francois, do you want to come back to your question?
Yes, so if there's no one else, yes. I'd like to know, you showed a graph where as you increase the number of CD3 zeta, you have, it takes longer to phosphorylate ERK. So is that only a model-based prediction, or is there also data confirming this prediction?
Yeah. So to our knowledge, there's not experimental data that validates that prediction. And it was on, our to-do list of something Jen wanted to follow up on and test and validate how changing expression of the CAR and specifically CD3 zeta would influence the ERK response, but we didn't get to it. But that's an important, I think, follow-on is to confirm or at least try to validate the model prediction. We haven't done that yet.
Super interesting prediction. Thank you.
Any other questions? I had one myself. I just, is there anything here in your modeling that is specific to a particular cancer type? Like, do you have anything that's specific to the kind of tissue that you're looking at that you would need to modify as you go to other solid tissues? I mean,
yeah. So the short answer is no. This is all, and especially right when we think about the tissue scale, which would be, I think, most relevant to a particular cancer type. It's agnostic to cancer type right now. But that's exactly what Colin is working on now. We have an experimental collaborator who's looking at breast cancer tumor slides in a mouse model. We also have been able to find in the literature some human samples for the distribution and spatial localization of immune and tumor cells in colorectal cancer. And that's a case where it's very interesting because there are certain structures or like levels of organization, especially at the periphery, so this invasive margin of a colorectal tumor, that I think would really complement the mathematical modeling that we're doing. So one question that he's trying to answer specifically for colorectal cancer is at that invasive margin, we see what are called experimentally and clinically, they see what are called tumor buds. So little clusters of cells, tumor cells and macrophages. And so a biological question is whether the macrophage is like recruiting tumor cells towards it or whether the tumor cell is there and then the macrophages are coming towards it. So we can answer that, right, by adding or removing cell-cell interactions in the agent-based model and see what gives rise to a spatial morphology that matches what's shown experimentally and clinically. So we're doing it, the short answer, both in breast cancer and then also in colorectal cancer. And the main things that we need to update are parameters related to the tumor microenvironment and like the growth rate or the proliferation rate of tumor cells to make them specific to that particular cancer type.
oh, fascinating. I think we've run to the end and the timing is very good. So let me say again, thank you for a really wonderful presentation. Lots of challenging things to think about here and lots of interesting directions to take these ideas. And I hope you are able to do so in the near future. And thank you so much for coming and giving that presentation here today.
Thank you. Thanks for the invitation. I'm excited to have this chance to talk with you all and happy to hear more feedback or answer